If I was to get points for that, yes.
We get points for pictures?
Huh?
We get points
Not
for
this
pictures?
semester.
No.
Not
Is
this
this not an art class?
But if Perry
Yeah,
Warner
this
wants, you
was-
would want a 3D model.
But you know,
on paper.
they do have biology classes, for drawing, so it's a biological, artistic type of class. I mean, who do you think draws a lot of these things in, our books? You got illustrators, right?
Yeah.
And these people have to practice on drawing animals and organisms and stuff. So if you are artistic, you can become an artist and just draw their stuff. Make a whole lot more money than I did.
I wasn't an artist. I wasn't an artist.
Well,
But then I quit that
why?
just
It's not too expensive.
because,
Nobody likes your art. Well, I mean,
It was subjectual.
yeah, they just tell me, hey, it's an abstract. Deal with it. Okie dokie. So that is your serial dilution. Number 5, describe in detail the process of prokaryotic DNA replication. Be sure to include, what else?
Terry, is that the one where, we're doing the double helix?
Hmm?
Like there's a double helix.
Well, yeah, what's going on inside that double helix, yes.
So, like, the hairpin and,
Yeah,
okay.
okay. So you want to talk about the proteins and the enzymes, everything that is involved, and their functions as well, okay? So, in order for DNA replication to start, where does it start from?
The DNA produces hydrogen bonds of the double helix.
So it does what?
It produces the hydrogen bonds.
Okay, but where does it start? Topoisomerases and gyrases. So how do they know where to start? Huh?
It starts right there at the protein molecule.
Okay, how and where does it know to start?
I just have to start enzyme.
Okay, so when you're talking about muscles in A and P, you have the origin and insertion, right? So the origin is where the muscle starts, or the non-movable form part of that joint is the origin, right?
Yeah.
Okay, well, it's the same thing. It's the origin of replication is where it's going to start. You have that down?
On another page.
Okay. So at the origin of replication is where it's going to start. So these topoisomerases will recognize that area and start to loosen the supercoil.
On these what?
It starts to unloosen the supercoil. The DNA double helix is in a supercoil at that point. It's really tight. So they're going to start breaking those hydrogen bonds that are between the nucleotides, right? So once those are being broken and loosened, it's going to loosen up that supercoil, allowing for the DNA helicase to enter in there. What's the job of the DNA helicase?
Unwind the DNA.
It starts to unwind that double helix, and it's going to create what we call a replication bubble. And on one end of that replication bubble, we have the replication fork, right? So when we're talking about what we're describing, we're just describing what's going on in one of those replication forks, okay? Fork. Like, ¿cómo se dice fork?
Tenedor.
Huh? Tenedor. Un tenedor. Un tenedor de replicación. Hay un enzyme helicase. Está, ¿cómo se dice unwind? Huh?
I don't know. I was gonna say, never mind.
You were gonna say this little quick.
Huh?
Oh, yeah,
Unzip.
deshacer.
Empecé?
Deshacer.
Deshacer. Okay, deshacer. As I said, ¿cómo se dice? Helix, duplica? Double helix, whatever. Anyways, so the helicase starts to create that replication fork, okay? And as it's unwinding, it's moving in one direction, right? And it's unwinding in one direction. What we know about how the DNA is replicated at this point, those two strands are now templates. So what does it mean to be a template?
It's the pattern for it.
Okay, it's the same thing as if I had a ink stamp and the pad is the template. I put it into the ink and I make my stamp. On the ink pad, it's one way, right? It's faced one way. It's actually opposite of what you would be looking at. But once I put it onto the ink, onto the paper, it's gonna look like I need it to look, right? So it's complementary. The two strands are complementary, so they are templates. So the same message that's going on this one is the same message that's going on that one. That's why you have complete copies of the DNA. But we also have to follow the rules. The rules state that it can only be added 5' to 3', right? And so, when I open up this bubble, that's why I have a leading strand, which is added 5' to 3' in this direction, right? But the lagging strand, you have to add it 5' to 3'. That would be going this direction. And you can't, because if I do this, as the helicase is moving away, all this other DNA is not going to be copied. So it has to go in a opposite direction, right? The 5' is actually over here. So I start 5', 3', 5', 3', 5', 3', 5', 3'. But I got to keep up with that helicase as it moves through and unwinds it, right? The leading strand is already following it by going continuously at it. But the lagging strand has to make Okazaki fragments, and it has to leapfrog and make an Okazaki fragment, leapfrog, make an Okazaki fragment, okay? So, let's talk about what enzymes are doing this. On the leading strand, you have a DNA primase. You can also call it RNA primase because it's laying down RNA, right? So there's a primase, that's an enzyme that's going to lay down a primer. What is the purpose of the primer?
Prevent hair strain.
Nope, not yet.
To facilitate the DNA nucleotide attachment.
To facilitate the DNA nucleotide attachment. So it basically has to have a handhold for something to grab onto to build the DNA strand, right? So this is going to happen in a continuous fashion because that DNA polymerase, which DNA polymerase is it? 3. DNA polymerase 3 will bind up to the end of that primer and start to lay down new nucleotides. It's going to do this in a continuous fashion. So it's very easy going. It's just going to
So then
continue.
nucleotides just go up?
The nucleotides are joining the polymerase is going here. So the nucleotides are joining it and it's creating a complete copy of that template. Okay? So that's going to happen in a continuous fashion. And
So
it's
is
gonna-
that when you have that little strip and you have all kinds of, you know what I'm saying? Like,
I think you're talking about transcription.
transcription.
It's not growing away, they're growing together.
Okay. But it
So-
has like all kinds of little rectangles and stuff.
Yeah,
That's.
if
that's
you were going to make
transcription.
rectangles the shape of your nucleotides, yeah.
Oh, okay.
Okay, you said DNA primase 3 binds to the end, and what does it lay down?
DNA polymerase.
Oh, well.
It's laying down a new strand of DNA and it's adding nucleotides in a continuous fashion. That's why we call it the leading strand, because it is going in a continuous fashion. Now, the lagging strand, that's a little bit different. Now, because we have to follow the rule going 5' to 3', when that DNA primase lays down a primer, DNA polymerase 3 will also attach onto there and lay down DNA, but only a short section of DNA, because that DNA primer has jumped and moved to another section and laid down a primer. Then the DNA polymerase 3 has to join that and lay down DNA. So it leapfrogs because that helicase is moving through and unwinding the DNA. So here's a DNA strand, as it's coming through, is opening up, right? And so that Okazaki fragment has to be laid down here, they jump over here, lay down this one here. So now you have a successive chain of Okazaki fragments. They're not bound together yet, right? So they cannot be a complete copy of that DNA, right? What has to happen in order to bind them together?
It have to be joined with DNA ligase.
So DNA ligase, another enzyme, will come in and weld them together. What happens to that primer? What gets rid of the primer? Because the primer is made up of what? RNA, right? So I got to get rid of that primer to have meaningful DNA put there.
Well, doesn't the polymerase 1 replace
DNA
RNA
polymerase
with
will
DNA?
come through and replace the RNA with DNA.
Nucleotide.
Yes.
Okay.
And so once that is done, now you have a complete copy of that DNA strand. And you have two copies because you have a template here on the leading strand and a template here on the lagging strand. Once that has completed, now you have two completely different copies of that DNA.
Can I read you mine that I have? Because I don't know, it's,
Sure.
and tell me if it's not the right word. I have at the origin of replication, DNA gyrase and
Topoisomerase.
yeah, whatever, loosen hydrogen bonds at the double helix. DNA heliocase unwinds the DNA helix and forms replication fork.
Mhm.
Stabilizing proteins stabilize strands, preventing hairpins. DNA primase creates RNA primers 5 to 3 on
Prime,
leading,
3 prime.
yeah, on leading and lagging strands.
Yes.
Leading strand is a continuous process. DNA polymerase 3 lays down nucleotides, creating covalent bonds. Polymerase 1 replaces RNA with DNA nucleotides. Lagging strands are formed headed from 4S Okazaki fragments joined with DNA ligase. Will that cover everything?
Did you mention DNA polymerase 1?
Mm-hmm.
Okay.
And polymerase 3.
Okay. All right. Yes. Okay, so, next one, describe the process of prokaryotic transcription from DNA to RNA. Be sure to include all enzymes and factors involved and their functions. Remember, it occurs in 3 different phases, right? So first off, you can say, well, prokaryotic transcription of DNA occurs in 3 phases. Initiation, elongation, and termination.
Wait, what did you say?
Initiation, elongation, and termination. So, when a transcript of DNA is going to be made, the RNA polymerase will have to read that strand of DNA in order to produce an RNA, right? So the RNA polymerase, along with what? Before that, what does the RNA polymerase carry along with it? A sigma factor, right? Sigma factor. The sigma factor, along with the RNA polymerase, will be able to then recognize an area on the DNA that's going to be transcribed. It's recognizing this because of